The end of chapter 13 is straightforward in one respect and a bit daunting in another. What it talks about is how metabolism is regulated by cells. One thing to notice is that the biochemistry of a cell is quite complicated. And this diagram here does a decent job in showing us a very small subset of reactions in biochemical systems. But key to all this is the role played by the process of glycolysis and the citric acid cycle in connecting different pathways together. Indeed, not all pathways are connected directly to glycolysis or the TCA cycle, but most of them are in a direct or indirect fashion. So these dots represent the substrates, and the lines connecting them represent pathways that are regulated by individual enzymes. So you can see that glycolysis can be utilized to feed in and out of alternative pathways by cells. If one concentrates on certain areas, one will notice that pyruvate has a very, very intense relationship with many other pathways inside a typical cell's biochemistry. It's not possible to deduce this, but from the previous diagram, some of those reactions are catabolic and other reactions are anabolic. But one thing is obvious, that everything is organized and regulated. Regulation means controlled. Organization means that it has its location and position within cells. The most common way of regulating biochemical pathways that we've discovered is by feedback regulation. So students need to think back to previous chapters of Bio 2, where we discussed positive and negative feedback, which is a common essential regulatory mechanism. The glycolysis and TCA cycle, respiration, is a great way to elicit how these mechanisms work. So studying how glucose is broken down and glycogen is involved and pyruvate is generated are areas of active biochemical research that has led to many findings about how cells can regulate the behavior of different pathways. And it turns out it's quite straightforward. There are key points in every pathway which are responsible for regulating that pathway. And this is beautifully illustrated with the example of glycolysis. So during glycolysis, there are at least 3 steps where the system can be regulated. And the most important ones of those are sometimes at places where energy is used. And this is one of those steps. So phosphofructokinase, and as we mentioned previously, you can decide what's going on there by the breakdown of this word. A kinase is something that adds phosphates to a molecule. Phosphofructose is normally the substrate, but to which this enzyme is adding something. So here is your phosphofructose, and here is your second phosphate group that's added by the enzyme. But this step here, if you ignore the rest of the slide, this step here is highly regulated. And it's very sensitive to the levels of ATP and ADP inside the cell. So there's some kind of mechanism that feeds back and allosterically regulates this enzyme. So allosteric regulation is the most common way in which enzymes themselves are activated and deactivated. And those enzymes then go on to activate and deactivate a biochemical pathway step. Now that we're familiar with how regulation takes place, let's now concentrate on the overall meaning of this slide. So the book talks a lot about glyconeogenesis. Glyco means glucose, neo means new, genesis is being made. So how can glucose be made by the cell if it doesn't come in as a food molecule? Well, it so happens that glycolysis can be run in reverse. And that's exactly what happens. So pyruvate can be converted back to glucose should the cell need that to power any other steps in this pathway. However, there are 3 steps that are energetically unfavorable for the reverse activity of every enzyme in glycolysis to be useful. And one of these steps is this one here. So where we go from fructose 6-phosphate to fructose 1,6, bisphosphate. And phosphofructokinase, let's get it right, is unable itself to be reversed because of a need for liberation of energy. So nature has sequestered another enzyme that's called fructose 1,6-bisphosphatase. And what it does, it removes the phosphate group from this molecule and establishes this molecule. So it goes in the reverse direction. And that enzyme is also sensitive to levels of ATP and ADP in the cell, but the reverse of this enzyme. So for this pathway, the reverse pathway, this enzyme is used, and for the forward pathway, this enzyme is used. When one enzyme is active, the other one should be deactivated, like a seesaw, teeter-totter. The textbook then moves on to talking about how reserves of energy are kept inside or around cells. So within cells, cells can store food molecules in special locations called reservoirs. And that's to prepare for periods when the supply of food itself, either through sunlight or from feeding, as in the case of animals, is between meals or during periods of inactivity like sleeping. And in animals, the most common form of storage molecule is glycogen. And what does glycogen look like? Well, it looks inside the cell as small granules that are hydrated with water and kept in the cytosol. And when magnified, glycogen is actually lots of glucose molecules linked together in a branching fashion. One would imagine that if glycogen is made of glucose, all you have to do is hydrolyze this link and you release free glucose molecules into the cell. But as we learned in a previous lecture, that is unwise, because that glucose would simply drift down through its gradient out of the cell into the environment. Remember, we're doing this in times of need, when glucose levels in the cell are very low. So to prevent that loss of glucose, nature, when it liberates glucose from glycogen, actually phosphorylates it straight into the second step of glycolysis, which is the glucose 6-phosphate. And the glucose 6-phosphate is unable to leave cells because it has no transporter. Every single branch point that you have can be attacked from its end by this enzyme. So the more branches you have, the faster the glucose can be liberated in times of need. And that's an important observation that we have made. In addition to storing glycogen as a starch, plant cells can also store oil droplets, as can animal cells. So these oil droplets can be visualized if they're stained. The oil droplets are dense areas of lipid storage that can be hydrolyzed and fed into the glycolysis cycle and the TCA cycle at various locations. The most common use of oils in plant cells is in the formation of seeds. So naturally, seeds are a reserve that has been designed by evolution to allow seeds to germinate for a period of time on these reserves as they develop their shoots and then their leaves. So without leaves and photosynthesis, seeds are unable to generate their own food. And animals have taken advantage of that as a source of food energy by eating seeds. Looking deep inside plant cells, we find that we have structures called chloroplasts. And here's a chloroplast surrounded by a bit of cytosol and then a cell wall of a plant. And here's a vacuole inside the plant, which is quite large. But let's focus on the chloroplast. Within the chloroplast, we have the stroma that carry out the process of capturing light. But embedded around them, we have two types of food storage. We have the carbohydrate starch granules, as well as the lipid droplets. And both of these can be utilized when the cell needs energy to power the photosynthetic pathways. We'll talk about those in the next chapter.